So I would like to use these two figures to help illustrate a problem that this raises when we think about the modern synthesis in biology. So we have two images here from some of the sources that were studying the Pitx-1 gene that causes the pelvis to form in the stickleback fish. And here's our sources if you're interested in those. One of the sources you've already seen as part of your group work, this one here. And I'd like to highlight kind of one of the things. This was an interesting study. It pretty elegantly showed they created probes that would dye the embryos blue in the regions where the Pitx-1 gene was being expressed, where the mRNA was being transcribed. They could identify that. And they noticed that in those fish that had their origins in lakes, there was no Pitx-1 expression, whereas those fish whose origins were from the ocean or whose parents had pelvic spines, they see Pitx-1 expression in the pelvis. And this was a pretty strong piece of evidence to indicate that perhaps this gene, this Pitx-1 gene, is actually the reason that pelvic spines are produced. And that's fine, except when you look at these images, you'll notice that it's not only the pelvis that expresses the Pitx-1 gene. Remember, the probes are targeting expression of this gene only. And so we see expression in the lips of the fish, but not only of the fish that come from the ocean populations, we also see it in the fish whose ancestors come from places where there are no pelvic spines. Well, that seems to contradict the idea that the gene for Pitx-1 creates pelvic spines, because if the gene for Pitx-1 is missing, thereby creating no pelvic spines, it should be missing from everywhere in the body where Pitx-1 genes are expressed. But we see that they're expressed essentially the same way in the mouth of both of these fish. And actually, if you were to cut these fish open and look at the inside, we would see Pitx-1 being expressed in other regions, in the pituitary gland, and other parts of the fish, and they're expressed the same in both of them. So that raises a problem. So on the one hand, it looks like the gene is responsible for the pelvic spine absence or presence. But on the other hand, the gene is not completely absent from one of the fish. It's only absent in certain places. So the modern synthesis can't really explain that. The modern synthesis would tell us, well, if the gene is absent, then we won't see any expression of that gene. And yet we do. We see expression in certain regions. So here's a follow-up study done a few years later, published a few years later. And this one takes a little bit more explanation, but in this study we have this FRIL fish, which are fish from a lake where the fish, from the ocean, where the fish contain the pelvis, with another species of stickleback, the LITC, which also contains a pelvis. So two different related three-spine stickleback fish, but from different populations, but both populations produce a pelvis. And then they take a population of stickleback, the Pitx-B stickleback fish, and that population of stickleback don't produce a pelvis. So as a control, they cross right here the FRIL population with the LITC population. You take two sticklebacks with a pelvis, and you get a stickleback with a pelvis. No big surprise there. In order to compare just how much of the Pitx-1 gene was being expressed in the FRIL population compared to the LITC population, that's what this graph down here shows. And in this case, the value of 1 is the value of 1, or the expression for the FRIL population, right? So this graph here is the expression in the FRIL population. Well, it's no surprise really that the LITC population is showing essentially the same amount of expression of the Pitx-1 gene, that it's normalized. Its value is also a value of 1, because both populations, the LITC and the FRIL sticklebacks, both of them have a complete pelvis. And we can see also that in their head, the expression is essentially equivalent. The error bars overlap with the value of 1, so we would be confident that this is essentially the same amount of Pitx-1 expression. Well, let's compare that over here to what happened on this side. So on the other side here, we took an FRIL fish, and it was crossed with a Pitx-B fish that has no pelvis. And they mapped how much Pitx-1 protein was expressed. And again, our graph is showing a normalized value of 1 for the FRIL fish, a fish with a pelvis. And we look at the expression of Pitx-1 in the head, and we see that it's essentially the same as the Pitx-1 expression in the head of the LITC fish and the FRIL fish. Basically, Pitx-1 is being expressed in the head at exactly the same rate in all of these fish. But look here, in the pelvic region, in the region of the pelvis, the Pitx-B fish is producing much less Pitx-1 protein, a significantly lower amount of Pitx-1 protein, and that's what we would attribute the lack of a pelvis to, this lack of Pitx-1. But now we have this problem. So Pitx-1 makes your pelvis go away. But Pitx-1 is also expressed in the head. It's in the lips, it's in the nervous system. So can the mutation that caused the pelvis to disappear, can that mutation have been in the Pitx-1 gene? Because a mutation in that gene that knocks the gene out so it's no longer expressed in the pelvis would mean that it would also knock out the expression in every other part of the body where it would be expressed. And yet, it appears to be expressed at a normal rate compared to a fish that has a complete pelvis. It's expressed at the normal rate in the head, even of fish that are lacking a pelvis. So this works in sort of direct contrast to what the modern synthesis of evolution would explain. The modern synthesis would tell us, well, hey, traits and variation in traits, like the kind of legs or spines or pelvis you produce, those variations come from changes to genes, and those changes to genes can be inherited. Well, in this case, the gene apparently changed in some parts of the animal's body, but not others. And yet, if the gene was changed, it should be inherited completely changed in the Pitx-B fish, and yet it's not. In some parts of the Pitx-B fish, this gene is normal, and in some parts of the Pitx-B fish, this gene is non-functional. And that stands in complete contrast to what Mendel taught us, to what all of the geneticists who studied the impact of genes on bodies explained to us. Which means we need a new explanation. And that's what the third digital notebook is going to help to try to resolve.